something very interesting, right? So this session is going to be taken in a very, in a different way, and it is definitely for all my first prof students. I'm your mentor at Unacademy, Dr. Reena Kaur, right? And being a, being a faculty and being a member committee in the curriculum. So I go more with the vertical and the horizontal integration concepts. So you know that nowadays, what is the type of question pattern the first MBBS is getting, right? MBBS first year may you are exposed to something which is called as ECE, that is your early clinical exposure, isn't it? So you might have often heard this term early clinical exposure from your teachers and from the faculties who are teaching you, isn't it? So we are going to look about what is this early clinical exposure. Basically, what we want is the clinical exposure right at the level of first MBBS. And this can be done either by taking you to the bedside, that is hospitals, or we can get graphs, or we can get, anything which will help you to correlate your physiology and to make the physiology more interesting. Another very important thing you see, which is coming nowadays, is a question which is definitely a clinical based question. So you're going to get typically a clinical based question. A clinical history will be given to you. Many a times you may be given a diagnosis, and sometimes you may even have to crack the diagnosis, right? So what we'll do, we'll have a clinical picture, and based on that clinical picture you get questions. So this is usually your 8 marks question, right? This is usually your 8 marks question. So today I was planning, or I have planned to take a question which is related to your general physiology. Good afternoon, boys and girls, Ugandar, and very good afternoon, beta. So let's check out how are you going to get a clinical question, which is 8 marks question with some history provided to you, and you will be asked to go through and to answer the questions. So I'm going to take such sessions, 2 to 3 sessions this month. Especially one of them I'm going to plan on endocrines, because endocrines is something where you get clinical history or a clinical based question. And second, I'm going to give a question that will be related to CNS, because these are the two, systems, your endocrines and CNS, where we are expecting clinical based questions for the first prof. Good afternoon, Jaswant. God, nahi, God, I'm not able to pronounce what's your name, so uh, इसीलिए, I just thought of, okay, voice is not clear. Let me check out. I think from my side things are all well. Okay, now let's come into a clinical question. A 43-year-old man is presenting to a physician with complaints of epigastric pain, right? Complaints of epigastric pain. And after a thorough workup, that means he has been investigated. The patient is diagnosed with peptic ulcer. Okay, the patient is diagnosed with peptic ulcer, and he has started medications. These medications are inhibiting the proton pump of the stomach. So because today I started that I'm going to take a question on general physiology. So the questions which I have taken will be related to general physiology. The same clinical question I can also take and put the question towards GIT. That is, I can take the question towards peptic ulcer. So this is totally depending upon the examiner, that whether I want to take a question on peptic ulcer, that is GIT, or I want to take a question on general physiology, right? Yes, Ujjwal, I'm going to take endocrines and CNS very, very soon. Hi, Ahmed, and hello, Aditya. I'm really glad to see all my first prof, which is my favorites among all the students being in the first prof. Yes, Ujjwal, let me plan this weekend only. I'm going to take something on CNS and endocrine, right? Yes. Now, a clinical history is being given to you. The question is 8 marks, and I'm now going to ask you, what is the proton pump that is referred here? One. Second is, what are the types of cell membrane responsible over here which are going to block, or the medication is going to block? Third important question which I can put on is, what are the four types of transport process across the cell membrane? Apart from this, if I want, I can tell you to draw a well labeled diagram, right? To draw a well labeled diagram of suppose sodium potassium pump, right? Or I can ask you, give me two examples of co-transport or any two examples of passive transport, or I can say you classify, right? I can say you classify. No, I'm going to explain, Ahmed. I'm going to explain now. So the first question is, what is the proton pump that is referred here? Right? So we are going to discuss all these questions. Now, these important questions are very, very important for general physiology, like the functions of the cell membrane, transport across the cell membrane, sodium potassium pump, GLUT, and simple diffusion. These are few of the questions which are a very, very, must known competencies for your first MBBS. Must known competencies. Out of this is in the form of an LAQ. These can even come as your SAQ or even sometimes just as a 2 marks BAQ. Apart from this, differentiate between GLUT and SGLT, that is a GLUT and a secondary coactive transport. You can just be given a 2 marks BAQ to differentiate between facilitated diffusion and secondary coactive transport mechanism. Right? Now, so if you want to summarize, this is the patient who is having peptic ulcer. Right? This is the patient of peptic ulcer, and the pump which we are speaking over here is the proton pump inhibitor. Right? The pump which we are speaking here is the proton pump inhibitor. This proton pump inhibitor is the sodium potassium ATPase pump, right, which is an example of primary active transport, which is an example of primary active transport, right? And other types of transport which we see are active and passive. So let's discuss first the cell membrane. Now remember, when we speak of the cell membrane, it is something very easy, right? It is something very easy, but definitely you need to have a detailed structure of the cell membrane nicely. This is called as a fluid mosaic model, okay? This is your fluid mosaic model, which was given by Singer's fluid mosaic model. In this model, the fluid is your lipids. Okay, the fluid is your lipids, and the mosaics are by the proteins. You know that it is a bilipid layer, right? It is a bilipid layer, and the lipids have got hydrophilic and hydrophobic portions. So it is just like this, like a pin. These are the heads of the pin, right? These are the heads of the pin, and this is the tail portion of the pin. This is the tail portion of the pin. The alignment is such that the tail portion of either side are facing each other, right? The tail portion of either side are facing each other, which are hydrophobic, and the tail ends are hydrophilic, right? The tail ends are hydrophilic. I don't know why it is echoing. The tail ends are hydrophilic. Now remember that this cell membrane has got a very, very important property, that it is semi-permeable, and also you can say that it is selectively permeable. When I say selectively permeable, I should know that it is going to allow only lipid soluble substances, gases, and steroids. Okay, lipid soluble substances, gases, and steroids. That means I must know, when I speak the steroids, I can also be asked a question related to hormone. Which hormone can easily pass through the cell membrane? So the hormones that can easily pass through the cell membrane will be only your steroid hormones, which is lipid soluble, and T3, which is lipid soluble, right? Your steroid hormones and your T3, they can only pass easily through the cell membrane, right? Now this fluidity means what? The membrane is kept in the fluid form, fluid gel like form. That depends upon the transition temperature, and the fluidity of the membrane is maintained by cholesterol. Right? The fluidity of the membrane is maintained by cholesterol. Now the mosaics are the proteins, and the proteins, very importantly, they show something which is called as lateral diffusion. Right? Lateral diffusion. The proteins are of two types. They can be peripheral proteins, which may be present either on the side of the cytosol, or they can be present here on the side of the ECF, and integral proteins, which are also called as the transmembrane proteins. The transmembrane proteins are all your pumps, your channels, your carrier proteins, your G protein. They are all examples of integral proteins. Right? Apart from this, the carbohydrate portion of glycoprotein, you have got something which is called as glycocalyx. This is very, very important because it is negatively charged, and because it is negatively charged, it is going to repel. And we often ask you that why the blood is free flowing. The blood is free flowing because the blood or the all the cells, they are having this negatively charged glycocalyx, because of which any cell can never get and come and stick to any blood vessel. So this glycocalyx is again very, very important. So you are going to get this cell membrane. You must remember this basic structure, that is the fluid mosaic model, where the fluid is the lipid and the mosaics are the proteins. Now this, classify the different transport mechanisms. I can give you as a 2 marks BAQ also. This can just come as a 2 marks BAQ. So if you are getting a 2 marks BAQ, you have to just write down. What I have experienced, my dear students, that you don't know what to write. If it is a 2 marks BAQ, you have to just write 1, 2, 3, 4 points. Don't elaborate, don't waste your time, because the paper has become very, very lengthy. There are 10, I think 10 BAQs you have to solve. So don't waste your time in 2 marks question. If you are asked, just tell me the types of transport processes. So active, passive, vesicular, and the transport across the epithelia. Right? So when we speak of the passive, simple facilitated diffusion, osmosis, this is your must know competency. Must know. You must know how to write this. Right? Obviously active is primary, secondary. Then there is vesicular transport, where we form a vesicle, exocytosis and endocytosis, and transport across the epithelium. What is this? Paracellular and transcellular. Okay, so when something is passing through the cell, this becomes transcellular, and when it is passing through the tight junctions between two cells, it is paracellular. Right? So again, an important 2 marks BAQ on the different types of transport processes. Now, this is again a very, very important 2 marks BAQ. What is the difference between simple diffusion and facilitated diffusion? You see, when you are getting the 2 marks BAQ, right, you definitely get this. Nowadays, I am seeing that difference between this and this. This is one of the favorite type of a question which is coming as a BAQ. So remember, when we speak of simple diffusion, there is no carrier. It does not show any saturation, because it shows a linear relationship. That means you keep on increasing the concentration, there is going to be increase in the diffusion. There is no saturation. There is no competitive inhibition. There is no competitive inhibition. There is no specificity. Whereas your facilitated diffusion shows something which is called as stereospecificity. That means if there is a particular carrier protein which is going to get attached to D isomer of glucose, it will not attach to L isomer of glucose. Apart from this, the rate of diffusion is faster in facilitated diffusion as compared to simple, which is slow. Right? Now the same thing, that is the difference between the simple and the facilitated diffusion, can also be shown in the form of a graph. See now, on x-axis you take concentration gradient, on y-axis you take rate of diffusion. So this is a linear correlation. That means you keep on increasing the concentration gradient, there will be increase in diffusion. There is no saturation. This is facilitated diffusion. This is a plateau which you are getting. So when you are getting a plateau, it is facilitated diffusion. This plateau, okay, this plateau is because of the saturation of carrier proteins. Okay, this is because of the saturation of the carrier proteins. So very common example is your glucose, right? Very common example is your glucose. Now let us see into, certain examples of simple diffusion. So remember that simple diffusion is going to be shown only by lipid soluble substances. Only by lipid soluble substances. Now, simple diffusion can be increased either by a pressure gradient or by a concentration gradient or by temperature gradient. Right? You can increase the simple diffusion. Problem with voice. I'll check it out. What is the problem? Where is the problem? Okay. Now, there are certain very important features of facilitated diffusion, right? Facilitated diffusion only takes place by a conformational change. Okay, it only takes place by a conformational change. That means the transport molecule, you can see it will bind to its receptor. Once it binds to its receptor, there is just going to be a change or a conformational change, right? So the carrier protein is going to bind to its site on the carrier, the substance to be transported is going to bind to a site on the carrier protein. As the substance binds to the size of the carrier protein, okay, as the substance binds as the substance binds to the site of the carrier protein, there is going to be a conformational change and this is going to release the substance. So simply a conformational change, right? Simply a conformational change and there is no other changes taking place. No ATP is required. This is the basic thing, that no ATP is required. Important examples are glutes and the urea transporters. They show facilitated diffusion. Remember, glutes is very important question. You have got only glut 4, which is insulin dependent, right? Which is present in your muscles and adipose tissue, okay, which is present in the muscles and the adipose tissue. So it is only the glut 4 which is insulin dependent, rest no other is, dependent. Mayur Ahmed, please carry on, try to concentrate on my words than my voice, because if I do any changes right now, I will have to stop the class. I will have to stop the class. So just concentrate on the matter, not on the voice. Okay? We have got one property in the synapse which is called as habituation. What is habituation? Ignorance. So just ignore the echoing of the voice and just concentrate on the matter. Life will be very simple if we accept that sometimes we have to ignore, right? Because if I do any changes, it is a live session and my class will stop. So it is very important that let's get connected. Okay, so just concentrate on the matter. Okay? Now, another important question which we get is difference between active transport and passive transport. This is again a 2 marks BAQ. Okay, it is again a 2 marks BAQ. 2 marks BAQ. Okay, it is again a 2 marks BAQ. Yes, my voice is echoing, but I think I'm able to understand everything. Now, active transport is active because it uses ATP. It uses ATP, one. Secondly, it is against the concentration gradient. It is against the concentration gradient. So because it is against the concentration gradient, it is called as uphill, right? Because it uses ATP, it is supposed to be a metabolic process. As compared to your passive diffusion where there is no use of ATP, it is along the concentration gradient. It is downhill because it is along the concentration gradient and it is simply a physical process. It is simply a physical process, right? So again a 2 marks BAQ to you. Now, primary active transport is primary. Why it is primary? Because it is causing direct breakdown of energy, right? Whereas why the secondary is secondary? The secondary is secondary because the transport is using the energy that is provided by the primary. I'll explain you this concept. Okay, I'll just explain you. The examples of primary are sodium potassium pump, the calcium pump, that is SERCA, which is the sarcoplasmic endoplasmic reticulum calcium ATPase pump, and the hydrogen pump. Secondary coactive transport, there are so many examples. You have got co-transport and you have got counter transport. Shri, I told you beta that the voice is echoing, but concentrate on the words. Right now I cannot make any change. Right now I cannot make any change because it is a live session. Now, examples of co-transport are, very important examples of co-transport are sodium bicarbonate, sodium chloride, sodium and glucose, that is SGLT, right? Then even there is sodium bile acids, okay, sodium bile acids. Examples of counter transport are, because there can come there can be a 2 marks BAQ also over the examples. Sodium hydrogen, then bicarbonate and chloride. Then even there is calcium sodium. The calcium sodium is present in the cardiac muscles. So remember, these examples of counter transport and symport are also important. Now, what is the difference between direct and indirect use of energy, right? So we'll just draw over here. What is the difference between the direct and the indirect use of energy? Now this is a sodium potassium pump which will always be present on the basolateral side, and this is going to be a luminal side, and suppose here there is presence of SGLT, that is sodium glucose co-transporter, which is going to take in sodium and also the glucose. The sodium potassium pump pumps out sodium and takes in potassium. It has got a coupling ratio of 3:2, that is 3 sodium out and 2 potassium inside, right? Now when it is doing this, it is causing hydrolysis. It is causing a direct hydrolysis of ATP. Therefore, it is called primary transport because it is causing a direct hydrolysis of ATP. Now, once it is pumping the sodium outside, what is occurring? Inside the cell, there is a decrease in the sodium concentration. That means it is creating a concentration gradient because it is keeping a low sodium inside. Okay, it is keeping a low sodium inside. Apart from that, sodium is a positive ion. That means it is also taking outside the positivity, that means it is also creating an electrical gradient. So it is creating two gradients, a concentration gradient and an electrical gradient, right? These two gradients are so strong that they allow the sodium to come inside into the cell with a secondary coactive mechanism. And when the sodium is coming inside, it can take along with it any other substance, which could be glucose, which could be amino acid, which could be galactose, or which could be any other ions. That means direct ATP is broken down by the primary sodium potassium ATPase pump, and it is creating a gradient. That gradient is utilized by the secondary coactive transport mechanism, and therefore it is called that this secondary coactive mechanism is using indirectly the energy. The main energy is broken down by primary. So this is the basic difference between your primary and secondary coactive transport mechanisms, right? Now, this is again a very important short note, that is sodium potassium pump, a very important short note. One of the high important competency for general physiology. You are also asked to draw a well labeled diagram. What points I can write here? I must know that it is a heterogeneous heterodimer. Okay, it is a heterogeneous heterodimer having two subunits, alpha and beta. The alpha subunit is actually the active one, because everything to bind is going to be present on the alpha subunit, and the beta is only meant for glycosylation. Right? On the inside, because you have to pump the sodium outside. So inside towards the ICF, you will have a sodium binding site, ATP site and phosphorylation site. And on the outside you will have potassium binding site, because you will have to bring the potassium inside into the cell, and this is QBank. What is QBank? It is nothing but it is actually your digitalis, which is supposed to be a cardiac inotropic agent, a cardiac glycoside. That means this is a drug which inhibits sodium potassium pump, and it is known to increase the force of contraction of the heart, which is called as inotropic effect. Right? Now, you must remember that this pump is stimulated by aldosterone, right, and even by insulin, and it is inhibited by ANP, that is atrial natriuretic peptide, and by the cardiac glycoside, that is digitalis, right? And what are the functions of this pump? Very important. It is contributing to your basal metabolic rate, because it is an energy consuming pump, and it is stimulated by the thyroid hormone. The thyroid hormone का जो important function होता है, क्या होता है? Calorigenesis, increasing BMR, that is brought about by increasing or stimulating this pump. And it is an electrogenic pump. Why electrogenic pump? Because it is creating or it is contributing to your resting membrane potential -4 millivolts. Because what is it doing? It is taking 3 sodium outside and getting 2 potassium inside. That means a net loss of 1 ion. Apart from this, it also maintains your cell volume, because if the pump fails, the sodium will accumulate inside, sodium is osmotically active, water will come inside, and this is how there can be swelling of the cells. So, sodium potassium pump is again a very important short note, including its diagram. What is this? This is your example of secondary coactive transport mechanism, that is your SGLT. you can see that sodium and glucose both are binding to a common site. Here, sodium is moving along its concentration gradient and glucose is being taken against its concentration gradient. Good afternoon, Anushka. Good afternoon, beta. Both of them will bind to the receptor and they will be released. Something very interesting for my first prof, this SGLT1 and 2 which are present in the intestine and in the PCT of the kidney, they are insulin independent. They are insulin independent. That means they do not require insulin, right? They do not require insulin. This also comes as an MCQ. That means in the kidney and in the intestine, the glucose reabsorption does not take place, does not require insulin. Symporters like sodium, amino acid, sodium, glucose, we've already seen, right? So you can see how beautifully this is the antiporters, where one substance will be taken inside and the other will go outside. These are vesicular transports, exocytosis and endocytosis, right? Exocytosis taking the things out, endocytosis taking sometime inside. Now, why is sugar added to ORS? It enhances acceptability, enhances taste, shell life or enhance salt absorption. Anybody would like to take a correct answer? किसी को बोलना है what would be the answer? Yes. So the correct answer is D. Because we know that glucose needs sodium for its reabsorption. So when you are adding glucose to ORS, this glucose will also take sodium and there will be reabsorption of both glucose and sodium. Both of them will create an osmotic effect and water will be reabsorbed. Okay? Water will be reabsorbed along with it. So just by adding sugar into ORS, you are increasing the reabsorption of salt as well as water. Which of the following is true about SGLT1? It is secondary coactive transport in brain, intestine, prostate or rods and cones. So remember, SGLT1 is in your intestine and the SGLT2 is in the PCT of the kidney. What is correct about your antiporter? Both molecules go in, one in, other out, both go out. One molecule goes in and two out. So remember, it is one molecule which is be going in and the other which will be going out. So this is all for today's session. The next session which I'm going to plan this weekend possibly or next will be a clinical based question, and I'm going to take either endocrines or CNS. Or we can, I think, discuss two cases. Yes, Samuel. Samuel, answer is B, beta. One molecule will go in and the other will go out. We can also discuss two cases. So I will discuss two cases. One will be from endocrine and one will be from CNS. Similarly, a clinical based history will be there and what related questions that are going to come. A typical SI में दूंगी so that you are able to solve your 8 marks question. Right? So with this, all my first prof, be very good and hardworking because your exams are approaching. You can provide me any suggestions if you have. If you want that, ma'am, take a particular section, you can always tell me. You can just leave a comment over here to any system, any topic you want me to teach you. Right? So let me know that whether the session was informative so that I can keep on adding up to your knowledge. So bye-bye, and we are present here at Unacademy to help you out in any and all form. So bye-bye and thank you very much.